lecturer or every GPAT class, they used to revise, they used to try to revise as much as they can so that you will effectively solve the paper. तो आपको जितना हो सके उतना रिवाइज करने के लिए ही जीपैट का क्लास होता है आपका. जीपैट का क्लास का मतलब ये नहीं कि आपको पूरा का पूरा आपका क्लियर हो जाए. कि ऑब्वियसली कुछ सब्जेक्ट ऐसे होते हैं कि उनको क्लियर करना जरूरी है, जैसे ऑर्गेनिक केमिस्ट्री, फार्माकोलॉजी. सो on those subject, obviously we will try to clear C or single basic also. There are certain subjects which have minor importance in your GPAT. For example, microbiology. They are only going to ask you 3 or 4 questions. So for 3 questions, it is a donkey work to get thoroughly regarding all chapters. So that's why we are going to see only important points which they have already asked frequently. We'll see the important point from microbiology. This is important. There are few chapters which are very important. What do you mean by microbiology? So obviously it is a study of pathogenic microorganism, which may be either bacteria, viruses, fungi, and all the rickettsia, fungi, viruses. So obviously remember, okay. So study of bacteria is known as a bacteriology. Try to remember or write down. Study of fungi is known as a mycology. So already they have asked question. Study of fungus is known as a mycology. Okay. Then study of bacteria, bacteriology. Study of virus, virology. Study of fungus, mycology. Or study of rickettsia is known as a phycology. PHYCOLOgy. Study of rickettsia is known as a phycology. There are some milestones in the history of your microbiology that you should know. जैसे यहाँ पे कुछ मैंने लिस्ट किया है. There are a lot of milestones they have mentioned in the books, but which are important I have listed over. So first one, in the 18th century. In the 18th century, first development of your smallpox vaccine was done by Edward Jenner. क्वेश्चन इसके ऊपर आ चुका है. What is the contribution of your Edward Jenner? एडवर्ड जेनर का कंट्रीब्यूशन क्या है माइक्रोबायोलॉजी में? कंट्रीब्यूशन ऑफ एडवर्ड जेनर, दैट इज नथिंग बट द एडवर्ड जेनर वाज डेवलप्ड और डिस्कवर्ड द स्मॉल पॉक्स वैक्सीन. स्मॉल पॉक्स जो होता है आपका, उसको वैक्सीन किसने इन्वेंट किया था? एडवर्ड जेनर. ट्राई टू रिमेंबर दिस. सो फर्स्ट, ओके, 18th century. In the 18th century, Edward Jenner, he discovered the discovery of smallpox vaccine. Okay, contribution is nothing but the discovery of smallpox vaccine. Then Robert Koch. So he discovered the anthrax disease. Anthrax. Okay, remember in 19th century, 19th century. Okay, so Paul Ehrlich, he developed the modern concept of chemotherapy and chemotherapeutic agent, means antibodies. Okay, so he is also known as a father of chemotherapy. Remember, Paul Ehrlich is also known as a father of chemotherapy. यहाँ पे इसके ऊपर भी important है. He is also known as a father of chemotherapy because he studied the various microorganisms. He developed the modern concept of chemotherapy and chemotherapeutic agent. So Paul Ehrlich is known as a father of chemotherapy. In 20th century, this August van Wassermann, he developed the complement fixation test for syphilis. Okay, and that's why this Wassermann test is a test used for the diagnosis of syphilis. syphilis के लिए कौन सा टेस्ट होता है? Wassermann टेस्ट, क्योंकि ये Wassermann ने complement fixation टेस्ट उसने इन्वेंट किया था. So in 20th century, August von Wassermann, he developed the complement fixation test for syphilis, and that's why that is a diagnostic test for syphilis. So already they have asked 2-3 times question. So what is the diagnostic test for syphilis? So that is known as the Wassermann's test. Okay, Wassermann's test. Okay, next is nothing but the Frederick Toward, he developed the bacteriophage. What is bacteriophage? When bacteria is attacked by virus, जैसे वायरस जब बैक्टीरिया के ऊपर अटैक करता है, सो वो बैक्टीरिया को अटैच होता है, उसके बाद में पेनिट्रेट करता है, पेनिट्रेट करने के बाद में उसका डीएनए उसमें छोड़ता है. ठीक है, सो व्हेन वायरस अटैक बैक्टीरिया, दैट इज नोन एज अ बैक्टीरियोफेज. सो दैट विल डिस्ट्रॉय द बैक्टीरियल सेल. When virus attack bacteria, that is known as bacteriophage. So it was discovered by Frederick Toward. Then Robert Cook. Remember, Robert Cook was the first who given the germ theory for disease. And there are postulate known as Cook's postulate. ठीक है, इसके कुछ postulates होते हैं. उसको बोलते हैं कुक्स पोस्टुलेट। कुक्स पोस्टुलेट मतलब उसने यही बोला है कि डिसीज जो है मोस्ट ऑफ द क्यों होते हैं ड्यू टू पैथोजेनिक ऑर्गेनिज्म एंड ऑल दैट। ठीक है, वो आपके माइक्रोबायोलॉजी में है। बट रिमेंबर जर्म थ्योरी ऑफ डिसीज वाज प्रपोज्ड बाय रॉबर्ट कुक। देन अगेन एरलिच इन 1992, दैट इज पॉल एरलिच, ही डेवलप्ड द थ्योरी ऑफ इम्यूनिटी। देन रिमेंबर इसके ऊपर एक क्वेश्चन आ चुका है, हु डेवलप्ड द माइक्रोस्कोप। सो रिमेंबर एंटनी वैन लीवनहुक। Anthony van Leeuwenhoek, he developed the microscope and he was the first to observe a microorganism under the microscope. माइक्रोस्कोप के नीचे माइक्रो ऑर्गेनिज्म कैसा दिखता है वो पहले इसने किसने एंटनी वॉन लिवनहुक। सो इसके ऊपर क्वेश्चन आ चुका है, हु डेवलप द फर्स्ट माइक्रोस्कोप, हु इन्वेंटेड द फर्स्ट माइक्रोस्कोप। सो एंटनी वॉन लिवनहुक। अलेक्जेंडर फ्लेमिंग इन 1928 ही डेवलप्ड द पेनिसिलिन्स। जो पेनिसिलिन आजकल अवेलेबल है इंजेक्शन के लिए, यूज के लिए, एंटीबायोटिक्स। सो दिस पेनिसिलिन्स वाज डिस्कवर्ड बाय फ्लेमिंग, अलेक्जेंडर फ्लेमिंग। यहाँ पे एंटनी वैन लीवनहुक, यहाँ पे अलेक्जेंडर फ्लेमिंग। दे डेवलप्ड द पेनिसिलिन, इन्वेंटेड द पेनिसिलिन। नाउ नेक्स्ट इज नथिंग बट द structure of DNA. Structure of DNA or structure of genetic material was discovered or was proposed by Watson and Crick. It is known as the Watson and Crick model. So Watson and Crick in 1953 they discovered the DNA structure. So these are some milestones in the microbiology. There are a lot of milestones, but you have to remember at least these. कम से कम ये तो याद रखो आप, मिनिमम ये आपको याद ही रखना पड़ेगा, इससे ज़्यादा बहुत सारे हैं। Now it's various microorganisms and disease caused by them. There are certain diseases they used to ask, syphilis कौन करता है, what is the causative organism for syphilis, causative organism for gonorrhea, like that. Okay, so microorganism known as bacteroids, okay, they causes the pelvic organ infection. So whatever pelvic organ infections are there, most of them are caused by these bacteroids. तो पेल्विक ऑर्गन इन्फेक्शन मतलब क्या, जैसे यूटीआई इन्फेक्शन, यूरिनरी ट्रैक्ट इन्फेक्शन, या फिर आपका जेनिटल इन्फेक्शन, सो ऑल जेनिटल ट्रैक्ट इन्फेक्शन, सो ऑल दैट आर कॉज्ड बाय बैक्टीरियाज। नाउ ब्रुसेला अबॉर्टस दे कॉजेस ब्रुसेलोसिस, रिमेंबर ब्रुसेलोसिस। देन क्लोस्ट्रीडियम टिटनी, इट कॉजेस द टिटेनस। ठीक है, आप कहीं अगर आपको कुछ लोहे का लग गया, मतलब लोहे की वजह से कोई आपको स्किन डैमेज हो गया है, फिर लोहे की वजह से कोई वुंड हो गया, सो एट दैट टाइम डॉक्टर यूज्ड टू इंजेक्ट दिस टिटेनस टॉक्सोइड, which is often from tetanus, Clostridium tetani, because that tetanus is caused by Clostridium tetani. Remember, now E. coli is responsible for the like infection of GIT, gastrointestinal infection like diarrhea and all that. UTI, urinary tract infection. So 90% of UTI is caused by E. coli. Then peritonitis. So that is infection of internal organs. Then again Mycobacterium leprae will cause leprosy. And remember this leprosy is also known as Hansen's disease. Leprosy is known as Hansen's disease. Okay, Mycobacterium leprae. And this mycobacterium is nothing but the acid fast bacilli. Remember it is a acid fast bacilli. ठीक है, इनके लिए एसिड फास्ट ट्रेनिंग होता है, सो अपन देखेंगे बाद में, सो विल सी इट अगेन। सो बट रिमेंबर ओवर हियर, सो माइकोबैक्टीरियम लेप्री एंड माइकोबैक्टीरियम ट्यूबरकुलोसिस both are the acid fast bacteria, acid fast bacilli. So they stained by acid fast staining. Okay, remember question आ चुका है, one of the following is known as acid fast bacteria. So माइकोबैक्टीरियम जो है, माइकोबैक्टीरियम लेप्री, माइकोबैक्टीरियम ट्यूबरकुलोसिस, so these are nothing but the acid fast bacteria. So Mycobacterium leprae will cause leprosy. Mycobacterium tuberculobacilli will cause tuberculosis. Tripomia pedilium will cause syphilis. So syphilis is the sexually transmitted disease. Syphilis is a STD. So this is caused by Tripomia pedilium. इसके ऊपर कम से कम 2 बार क्वेश्चन हो चुका है. So what is the causative organism for syphilis? Tripomia pedilium. Triponema pedilium. Okay, Triponema pedilium. So that is a causative organism for syphilis. And diagnostic test क्या है इसके लिए? We have seen in the previous slide. Wasserman's test is used for the diagnosis of this syphilis. उसके ऊपर भी क्वेश्चन आ चुके हैं. Such kind of things. क्वेश्चन कहाँ से आता है और रिपीट हो सकता है कि 100% रिपीट हो सकता है. They will ask you in a twisting or turning. they can ask you the question by twisting or turning. So those points are very very important. So remember again over here, थोड़ा सा जल्दी आते जाओ आप, आपका टाइम दिया है मैंने. So remember, most of the infection of UTI, GIT and peritonitis is caused by E. coli. Okay. Then your Mycobacterium leprae, acid fast bacteria will cause leprosy. Okay. Also known as a Hansen's disease. Then Treponema pallidum will cause syphilis. Okay. It is a diagnostic test for syphilis is nothing but the Wassermann's test. Okay, Wassermann's test. Now Vibrio cholera will cause cholera. Vibrio cholera will cause cholera. Cholera or it is known as a diarrhea, watery diarrhea. So in which there is a watery rice stools. Okay, cholera में क्या होता है? Cholera का infection हो गया तो इतना डायरिया होता है कि cholera से आदमी नहीं मरता. आदमी मरता है due to dehydration. क्वेश्चन उसके ऊपर भी आ चुका है. So in the cholera what happens? This infection Vibrio cholera will cause it is an intestinal infection. So it will cause intestinal infection and it will lead to a watery diarrhea, watery rice water diarrhea. Okay, rice water diarrhea. So they already asked the question. Rice water diarrhea is a characteristic of one of the following disease. So cholera is nothing but the rice water diarrhea. बहुत सारे, this was a very big epidemic. It was a very big epidemic. Coronavirus is nothing. So this cholera was a very big epidemic and a lot of mortality was there due to this cholera. Now, what is the mortality for corona? It is just a 1%. But cholera में ऐसा था कि चलते चलते आदमी को डायरिया हो जाता और वो चलते चलते मरता. So it was very big pandemic. Cholera, Vibrio cholera. Remember, rice water diarrhea is the characteristic. Salmonella typhi will cause typhoid fever, obviously. Rocky Mountain spotted fever is caused by rickettsia. Then Prototella protosis will cause whooping cough. Question इसके ऊपर भी आ चुका है. What is the positive organism for whooping cough? Okay, whooping cough उसको मराठी में बोलते हैं टांग्या खोखला. Whooping cough is caused by Prototella protosis. Prototella protosis. Okay. Then this Chlamydia trachomatis is responsible for the venereal disease. Then diphtheria is caused by Corynebacterium diphtheria. Tuberculosis is caused by Mycobacterium tuberculosis. Most of the peptic ulcer is caused by Helicobacter pylori. Gonorrhea is caused by Neisseria gonorrhea. Neisseria. Then plague is caused by Yersinia pestis. इसके ऊपर भी क्वेश्चन आता है. Positive agent for plague, Yersinia pestis. Then heart infection, endocarditis. Heart infection मतलब क्या? Endocarditis or simply pericarditis or endocarditis. It is mostly caused by Streptococcus viridans. Streptococcus viridans. तो आपके इसमें बाकी माइक्रोबायोलॉजी के बुक में दिया है क्लासिफिकेशन ऑफ बैक्टीरिया, shapes of bacteria, then hook shaped, rod shaped. So that classification is not much important, but if you can read, that is very good. Now yellow fever is caused by this flavivirus. Yellow fever is caused by flavivirus. Now this flavivirus is again responsible for the, or it is a causative organism for dengue. जो डेंगू होता है आपको, वो डेंगू भी इसकी वजह से होता है, flavivirus. So remember these. It is very simple names of microorganism and their disease caused by them. Different types of microscope. So they have already asked first type of microscope. First type of microscope is nothing but the ordinary light microscope. First one is the light microscope. Microscope बहुत सारे प्रकार के होते हैं, जैसे light microscope, transmission electron microscope, scanning electron microscope. ठीक है, सबसे ज़्यादा resolution scanning electron microscope होता है ACM. So light microscope, what is the comment? If you want to see in the detail what are the parts of eyepiece, the condenser, okay, eyepiece, condenser, lens, etc., etc., that is mentioned in the book. So they are not going to ask question on this. So what is the resolution obtained by light microscope? उसके ऊपर क्वेश्चन आ सकता है. So what is the resolution? Maximum resolution is 0.2 micrometer. Resolution means what? It is the capacity to distinguish between two close structures. Maximum resolution is 0.2 micrometer. Remember. Now commonly employed light microscope is nothing but the headlight microscope, bright field or dark field or phase contrast and fluorescence microscope. These are the types. लाइट माइक्रोस्कोप के टाइप कितने हैं? कितना है? ब्राइट फील्ड है, डार्क फील्ड है, फेज कॉन्ट्रास्ट है, फ्लोरोसेंट माइक्रोस्कोप है. नाउ क्वेश्चन इसके ऊपर एक बार आ चुका है, एक नहीं दो बार आ चुका है. ओके, सो यू आर यूजिंग सीडर वुड ऑयल. You are using cedar wood oil for magnification. So what is the magnification that is provided by cedar wood oil? So 100 times magnification, 100x. So question इसके ऊपर आ चुका है. So cedarwood oil will provide a magnification in the magnification dash dash. ऑप्शन में ए में आएगा, ए ऑप्शन आएगा 10x, दूसरा आएगा 25x, तीसरा आएगा 100x, चौथा आएगा 500x. So magnification given by cedarwood oil is a 100 times means 100x. Okay, so times को x भी बोलते हैं. So 100x cedarwood oil will give 100 times magnification. मतलब एक स्ट्रक्चर जो दिख रहा है उससे 100 गुना ज्यादा नजदीक दिखेगा वो. कौन सा? बिकॉज ऑफ cedarwood oil. ओके, रिमेंबर एक क्वेश्चन इसके ऊपर आ चुका है. Now second type of microscope is a transmission electron microscope. So again it will give very good resolution, fabulous resolution. Fabulous means very good. Fabulous resolution of 0.5 nanometer. So it can distinguish the object which are at a distance of 0.5 nanometer. तो 0.5 नैनोमीटर के नजदीक नजदीक अगर दो दो स्ट्रक्चर है तो वो भी आपको ट्रांसमिशन इलेक्ट्रॉन में दिख जाएंगे. वेयर इन द ट्रांसमिशन इलेक्ट्रॉन माइक्रोस्कोप very short wavelength, okay, very short wavelength direct electron beam is used for magnification. इसका characteristic है याद रखो. Third, differential interface contrast microscope. So to obtain a 3D and colored images, this microscope is used. Differential interface contrast microscope. Then next one is scanning electron microscope, most advanced than transition electron microscope. So obviously सबसे ज्यादा resolution किसका होगा? स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप पर सबसे ज्यादा रेजोल्यूशन होता है. रिमेंबर दिस पॉइंट्स. As I have told you, there are different classes of bacteria. Some okay. So if you want to, you can read from book. The questions they have already asked from these parts. Types of bacteria. Okay, depending upon their optimum pH for growth. कौन सा बैक्टीरिया किस pH पे मीडियम का pH होना चाहिए तब उसका ग्रोथ है. So the pH, maximum pH at which bacteria will show highest growth, that is known as the optimum pH value for microbial growth. So based upon the optimum pH value, bacteria they are classified into three types. What is the optimum pH of bacteria? So the pH of medium at which they will show there is a highest growth of bacteria, highest rate of growth. So the pH of medium at which bacteria will show highest rate of growth, okay, that is known as the optimum pH. Okay. Now based upon that, bacteria are classified into three types. First one is known as acidophiles. Acidophiles मतलब क्या है? Obviously acid मतलब क्या है? कम pH. So these are the bacteria which can grow at a lower pH. So acidophils will grow at a pH 1 to 6.5. For example, Thermobacillus theothixant. Thermobacillus theothixant. Okay, so it will grow at a pH 2 to 3.5. Okay, so acidophils कब ग्रो कहाँ पे कौन से pH पे ग्रो होते हैं? pH 1 से लेके 6.5 तक उनका ग्रोथ होता है. फॉर एग्जाम्पल दिस थियोबैसिलस थियोथीक्संट थियोक्सिन. So this Thiobacillus thiocxan, it will grow at a specific optimum pH, that is 2 to 3.5. Then Lactobacillus acidophilus is the name of bacteria. Lactobacillus acidophilus, it will grow at a pH, optimum pH is nothing but the 5.8 to 6.6. They can ask you question, they will give you the examples of bacteria, and they will ask you to identify the acidophilus bacteria. Okay, acidophil. Neutrophils means was those bacteria which has optimum pH in a neutral range. So like neutrophils means what? They will grow at a pH 6.5 to 7.5. न्यूट्रोफिल्स में जितने भी बैक्टीरिया आते हैं उनका हाईएस्ट ग्रोथ कब होगा? जब पीएच मीडियम न्यूट्रल होगा. सो न्यूट्रोफिल्स विल ग्रो at a pH 6.5 to 7.5. Examples. What are the neutrophils examples? याद रखो. So examples are nothing but the E. coli, Salmonella typhi, and other pathogenic bacteria. तो example याद रखो. E. coli, Salmonella typhi. Those are nothing but the neutrophils. Acidophils, neutrophils, basophils. Basophils का क्या करेंगे? So basophils will grow optimally. pH of medium is a basic. Based upon that optimum pH. What is the optimum pH? pH at which bacteria will show highest growth rate. The pH at which bacteria will show highest growth rate, and depending on that, bacteria are classified into three types. So acidophils, basophils, and neutrophils. Acidophils means what? They will grow at an optimum pH of 1 to 6.5. For example, Thiobacillus thiocxan. So that will grow at a 2 to 3.5 pH. Lactobacillus acidophilus will grow at a 5.8 to 6.6. ये ऑप्टिमम होता है, ये pH पे उसका सबसे ज्यादा ग्रोथ रेट होता है, सबसे ज्यादा मल्टीप्लिकेशन होता है. So neutrophils means what? Neutrophilus means those bacteria which will show highest growth rate at a pH 6.5 to 7.5. pH रेंज में 6.5 7.5 के रेंज में अगर बैक्टीरियाज मल्टीप्लाई कर रहे हैं ज्यादा, उनको बोलेंगे न्यूट्रोफिल्स. फॉर एग्जांपल ई कोलाई, साल्मोनेला टाइफी एंड अदर. बेसोफिल्स और अल्केलोफिल्स. तो बेसोफिल्स थोड़ा सा यूज नहीं करते टर्म, बिकॉज बेसोफिल्स WBC भी होते हैं. So that's why alkalophils. Okay, alkalophils. So these are the bacteria that will grow at this optimum pH, that is 7.5 to 14. For example, Vibrio cholera. So what is the optimum pH of growth for this bacteria? pH 9. So Vibrio cholera will show highest rate of multiplication at a pH 9. So depending on the pH, optimum pH, so bacteria are classified into these three types. इसके ऊपर क्वेश्चन आता है. सो मैच द फॉलोइंग टाइप, या फिर डायरेक्टली क्वेश्चन पूछेंगे. ई कोलाई इज न्यूट्रोफिल, याद रखो ई कोलाई न्यूट्रोफिल होता है, साल्मोनेला न्यूट्रोफिल होता है, जो वाइब्रियो कोलेरा होता है वो अल्केलोफिल होता है या फिर बेसोफिल. अगेन फॉर ग्रोथ ऑफ बैक्टीरिया यू नीड ऑप्टिमम टेम्परेचर. टेम्परेचर इज वेरी इंपॉर्टेंट फॉर योर ग्रोथ ऑफ बैक्टीरिया. जैसे आप इनोकुलेशन करते हो तो उनको एक पर्टिकुलर टेम्परेचर पे ही इंक्यूबेट करना पड़ता है, तभी जाके आपको ग्रोथ मिलेगा. What is the optimum growth temperature of bacteria? Optimum growth temperature of bacteria. So temperature range for bacterial growth. The highest temperature at which there is a highest rate of bacterial multiplication, or the highest temperature which allows the rapid growth during short period of, that is known as the optimum growth temperature of bacteria. Bacteria क्या है कि उनके temperature भी important है. एक ऐसा temperature होगा उसमें क्या होगा, बैक्टीरिया का सबसे ज्यादा ग्रोथ होगा. That is known as the optimum temperature, optimum growth temperature. Temperature range for bacterial growth. The highest temperature which allows the rapid growth and multiplication for a short period, that is known as the optimum growth temperature of your bacteria. So depending on that also bacteria are classified. There are different way to classify the bacteria, but particularly these two I have taken because they have already asked question on this. So group growth temperature and examples. Okay, growth temperature and examples. What is obligate psychrophiles? Obligate psychrophiles. So these obligate psychrophile bacteria, they will grow at a temperature. So they can grow at a temperature 0 to 5 degrees Celsius. That is the minimum temperature. So they can survive at a 0 to 5 degrees Celsius also. They can survive. But optimum temperature for their growth is nothing but the 10 to 15 degrees Celsius. They will grow maximum at a maximum rate, highest rate at a 10 to 15 degrees Celsius, that is known as obligate psychrophiles, and they can tolerate maximum temperature known as 20 degrees Celsius. For example क्या है उसका, Vibrio pyroerythrus, Vibrio psychroerythrus, Vibrio psychroerythrus. So this is known as obligate psychrophiles. Psychrophiles देखो psychroerythrus, psychro को जोड़े लगाओ. Obligate psychrophiles, minimum temperature क्या है, they can survive 0 to 5 degrees Celsius. Optimum temperature at which they will show highest growth, that is 10 to 15. Maximum temperature that they can tolerate, that is 20 degrees. And facultative psychrophiles, they can survive at the same point 0 to 5. Okay, then optimum temperature for their growth is nothing but the 25 to 30, and they can tolerate 35 degrees Celsius. For example, Pseudomonas fluorescens. So they can ask question, one of the following organism is a facultative psychrophile, or one of the following organism is a mesophile or facultative thermocol, etc. Obligate. That's why this is a very important. But next category is nothing but the mesophils. Mesophils, these are the bacteria which they can tolerate or they can survive at 15 to 20 degrees Celsius. Their optimum temperature for growth is nothing but the 35 to 40 degrees Celsius. They can survive maximum temperature 65 degrees. For example, Corynebacterium diphtheria. Corynebacterium diphtheria. This is the mesophil. Now next one is a facultative thermocol. Am I audible you clearly? आवाज आ रहा है सगळा? आवाज येत आहे का? Facultative thermocol. So these bacteria, they can survive at 35 to 40 degrees Celsius. Minimum temperature for growth minimum temperature required for growth is 35 to 40 degrees Celsius. But optimum temperature मतलब क्या? So they will show highest rate of their multiplication at a 50 to 55 degrees Celsius. And maximum they can tolerate 65 degrees Celsius. Example is nothing but the Streptococcus thermophilus. Streptococcus thermophilus. And last is nothing but the obligate thermobiles. Obligate thermobiles. Okay. So minimum temperature for their survival is a 50 to 55 degrees Celsius. Minimum temperature 65 to 70 degree. At this temperature, they will show highest growth rate and they can tolerate 80 degrees Celsius also. Example is Thermus aquaticus. Example Thermus aquaticus. Remember from this organism is one very important enzyme that is isolated. It is very important for your polymerase chain reaction. Tell the name of that enzyme. इसमें से एक एंजाइम निकलता है थर्मस एक्वाटिकस से वो एंजाइम आपके पॉलीमरेज चेन रिएक्शन के लिए बहुत ज्यादा इंपॉर्टेंट होता है उसका नाम क्या है इज नोन एज अ टैग डीएनए पॉलीमरेज टैग डीएनए पॉलीमरेज इज ऑब्टेन्ड फ्रॉम थर्मस एक्वाटिकस रिमेंबर when we'll see that biotech part at the time I will also explain there are different types of tag DNA polymerase. Tag DNA polymerase is the enzyme that is obtained from Thermus aquaticus and that enzyme is stable up to 80 degrees Celsius. Because obligate thermophiles they are stable they can tolerate 80 degrees Celsius. If they can tolerate 80 degrees Celsius whatever enzymes they are obtained from these microbes that enzyme also can tolerate 80 degree. याद रखो उसको थर्मस एक्वाटिकस में से कौन सा एंजाइम आता है डीएनए पॉलीमरेज टी ए जी टैग डीएनए पॉलीमरेज टैग डीएनए पॉलीमरेज टैग डीएनए पॉलीमरेज इज यूज्ड इन द पॉलीमरेज चेन रिएक्शन एक स्लाइड के ऊपर कम से कम दो क्वेश्चन होंगे दिस पार्ट विल सी देयर आर डिफरेंट पार्ट्स ऑफ बैक्टीरिया ओके यू कैन सी इन दिस इमेज पार्ट्स ऑफ बैक्टीरिया bacterial cell obviously this bacterial cell will have different part like it will have cell wall it will have nucleic acid that is the chromosomes okay chromosomes nucleic acid then bacteria will have capsule okay capsule then then pili etc see one by one. So first one is nothing but the cell wall. This part is known as the cell wall. Cell wall. Cell wall is nothing but the outer covering of bacterial cell. So there are different kind of cell wall. There depending on this cell wall, bacteria are classified into two types: gram positive, gram negative. Gram positive bacteria will have thick cell wall. Gram negative bacteria will have thin cell wall. Cytoplasm layer will be less in the gram negative bacteria. Gram positive में layer ज़्यादा होता है. Okay, then plasma membrane. Plasma membrane is circular. Okay, inside the cell there will be DNA. Now there are two types of DNA in the bacteria. बैक्टीरिया में दो टाइप के डीएनए होता है फर्स्ट वन इज अ क्रोमोसोमल डीएनए क्रोमोसोमल डीएनए मतलब उसका मल्टीप्लिकेशन के लिए इंपॉर्टेंट होता है क्रोमोसोमल डीएनए chromosomes okay so chromosomal DNA and another DNA that known as extra chromosomal DNA so another kind of DNA is there known as extra chromosomal DNA extra chromosomal DNA का क्या फंक्शन है extra chromosomal DNA from the bacteria is known as a plasmids replicating extra chromosomal DNA. You can see here plasmids circular DNA okay. Circular DNA that is the extra chromosomal DNA so on that DNA on that chromosome there is no genetic information इसके ऊपर genetic information नहीं होता है so whatever genetic information of bacteria that is present in the chromosomal DNA okay so बाकी जो है self-replicating circular DNA is known as an extra chromosomal DNA and known as a plasmids whole recombinant DNA technology is based upon this plasmids vectors okay there are kind of vectors plasmid vectors okay cosmid vectors lambda phase vectors that is viral vector artificial vectors etc then what are the organs this purple part is known as a capsule this purple part is known as capsule this cytoplasm these positions on the capsule is known as a pili it is known as a pili or sex pili is known as a pili or sex pili. Its granules are known as ribosomes. You know what is the function of ribosome? Important for protein synthesis. I want to repeat it. It is nothing but this part. This purple color part is a capsule. So these are known as a flagella. Remember over here, these are the flagella. They are the organs of locomotion. Bacteria will move with the help of this flagella. बैक्टीरिया के मूवमेंट के लिए कौन सा ऑर्गन रिस्पॉन्सिबल है फ्लैजेला क्वेश्चन आ चुका है तो following part of bacterial cell is responsible for the locomotion of bacteria. Remember, flagella is the organ of locomotion. Okay, so bacterial cell, bacteria का cell, which consists of cell wall, cytoplasm, nuclear material, ribosomes, mesosomes, etc. Now, depending on the type of cell wall, they are classified into two types: gram positive bacteria and gram negative bacteria. So gram positive bacteria, gram negative bacteria, we have different cell wall and composition of cell wall as compared to each other. Depending on that gram positive, gram negative, we'll see the difference. Now another part is known as a capsule or slims. Capsule or slims. इसके बारे में क्वेश्चन ऑलरेडी पूछा है. What is capsule? So capsule is nothing but the outer envelope or outer covering of bacterial cell. So capsule is nothing but the some kind of or some sort of envelope present on the bacterial cell. Okay. If this envelope or this coating is contrarily a loose structure and which will diffuse into the surrounding medium, then that time this is known as slimes. बैक्टीरिया है आपका, बैक्टीरिया के ऊपर एक कवरिंग होता है, उसको बोलते हैं कैप्सूल. कैप्सूल क्या होता है कि रिलेटिवली रिजिड कवर होता है, रिजिड मतलब वो फिक्स होता है कैप्सूल. लेकिन स्लाइम्स मतलब क्या, स्लाइम्स मतलब कैप्सूल, स्लाइम्स मतलब क्या, बैक्टीरियल सेल के ऊपर थोड़ा थोड़ा ऐसा डिफ्यूज मतलब लूज लेयर होगा. इफ देयर इज अ लूज लेयर surrounding the bacterial cell, okay, that is known as a slimes. Now that loose layer may be diffused into the surrounding medium. कैप्सूल रिजिड होता है, स्लाइम रिजिड नहीं होता है, इट इज अ डिफ्यूजिबल. मतलब अगर जिस भी मीडियम में बैक्टीरिया रहेगा उसमें वो डिफ्यूज हो जाता है. स्लाइम का ही कैप्सूल बनता है बाद में. If envelope or coating is contrarily loose structure which often gets diffused into the corresponding medium, okay, it is known as slimes. Capsule and slimes, these structures helps to the bacteria to survive in the extreme condition. Extreme condition मतलब क्या है, favorable condition क्या होता है, जैसे optimum pH बोला है, optimum temperature बोला है, okay, then grow media है, उसके न्यूट्रिशन है, so that is known as a favorable condition. आपको अगर खाने पीने को सब कुछ अच्छा है, खाने पीने को सब कुछ अच्छा है, एसी में बैठे तो ऑब्वियसली वहाँ पे आपका ग्रोथ ज़्यादा होगा. आपका तो नहीं होगा, आप सो जाओगे, लेकिन बैक्टीरिया का ग्रोथ ज़्यादा होगा, ठीक है, क्योंकि दैट इज अ फेवरेबल कंडीशन. बट व्हाट इज अ एक्सट्रीम कंडीशन, सपोज आपको 50 डिग्री सेल्सियस का टेम्परेचर है, वहाँ पे काम करने के लिए बोला, दैट इज अनफेवरेबल कंडीशन. वहाँ पे पानी ही नहीं है, unfavorable condition. So these capsule or slimes helps in the bacteria to survive in the extreme condition, unsuitable condition. What is flagella? Flagella is organ for locomotion. Flagella is the organ for locomotion. Next one is the fimbri or pili. Fimbri or pili. Fimbri or pili. So fimbri or pili is also known as sex pili. They are known as sex pili because their synthesis is predominantly directed by or determined by sex factor. They are also having major role in the disease spreading in the humans. ये जो पिलाए होते हैं इसके से infection spread होता है. And पिलाए, they are also known as sex पिलाए because their synthesis is directed by sex factors. Next one is the different phases of your bacterial growth. बैक्टीरियल ग्रोथ के स्टेज कितने होते हैं, बैक्टीरिया का ग्रोथ किस किस स्टेज में होता है, क्वेश्चन इसके ऊपर भी आता है. There are 4 phases of bacterial growth. First one is a lag phase, second one is a log phase, third one is a stationary phase, and fourth one is a decline phase. So you might have seen these phases in your, you might have studied these phases in your college. आपको पढ़ाया होगा. So first lag phase, second log phase, third stationary phase, and fourth decline phase. So okay, जैसे आप बैक्टीरिया को इनोकुलेट करते हो, so if you make a medium and if you want to isolate the bacteria, so you isolate or you inoculate the bacteria in the petri plate or medium. So it will take some time to adapt the condition, it will take some time to adapt the environment, that is known as a lag phase, initial stage of adaptation. So there is only little growth of bacteria or there will be no growth of bacteria. Bacteria will try to adjust himself in the new environment known as a lag phase. Okay, now second phase is known as a log phase. So एक बार एडजस्टमेंट हो गया तो अपना अपना वंश आगे बढ़ाएगा, ठीक है. So that is known as a log phase. और एक्सपोनेंशियल फेज. सो रैपिड एक्सपोनेंशियल ग्रोथ ऑफ माइक्रोऑर्गेनिज्म. तो इस फेज में क्या होता है कि बैक्टीरिया मल्टीप्लाई होता है बहुत ज्यादा फास्ट. सो देयर इज अ रैपिड रेट ऑफ मल्टीप्लिकेशन एंड एक्सपोनेंशियल ग्रोथ ऑफ माइक्रोऑर्गेनिज्म. Okay, so this is known as a log phase or exponential phase. Now, third one is known as a stationary phase. Third one is known as a stationary phase. So, leveling of growth of microbes. Leveling of growth of microbes. Okay, straight line will be there. I will explain this in upcoming slides. वो होता क्या है कि एक्सपोनेंशियल ग्रोथ में वो मल्टीप्लाई होता है फास्ट. अभी जो मल्टीप्लाई हो रहा है तब सब कुछ न्यूट्रिएंट वगैरह सब कुछ है. अभी क्या हो रहा है थर्ड फेज में आता है स्टेशनरी फेज. तो वहां पे क्या है रेट ऑफ मल्टीप्लिकेशन इज इक्वल टू रेट ऑफ डेथ. तो रेट ऑफ मल्टीप्लिकेशन विल बी इक्वल टू रेट ऑफ डेथ ऑफ बैक्टीरिया एंड दैट्स व्हाई स्टेशनरी फेज. दैट्स व्हाई स्ट्रेट लाइन. लेवलिंग ऑफ ग्रोथ ऑफ माइक्रोब्स straight line due to depletion of nutrition. Now next phase is nothing but the decline phase. So decline in the growth of microorganism is because of the lack of nutrient and accumulation of toxic metabolites or toxic waste product. ठीक है. तो अभी बैक्टीरिया का ग्रोथ तो हो चुका है सब. अभी अभी न्यूट्रिशन खा लिया उसने सब. अभी उसको न्यूट्रिशन खाने के बाद उसको वेस्ट प्रोडक्ट भी डालेंगे. आप खाना खाने के बाद छोड़ते हो ना मॉडर्न मेटाबॉलिज्म वही रखते हो. वैसे वो टॉक्सिक मेटाबोलाइट टॉक्सिक वेस्ट प्रोडक्ट वो डालेगा मीडियम में. बिकॉज ऑफ दैट व्हाट विल हैपन देयर विल बी नो ग्रोथ ऑफ बैक्टीरिया. तो डिक्लाइन फेज. डिक्लाइन इन द ग्रोथ ऑफ माइक्रोऑर्गेनिज्म बिकॉज ऑफ द लैक ऑफ न्यूट्रिएंट एंड एक्युमुलेशन ऑफ टॉक्सिक प्रोडक्ट. इसके ऊपर क्वेश्चन आ चुका है. सो accumulation of toxic product and depletion of nutrition occurs in the one of the following phases of bacterial growth cycle. ऐसा क्वेश्चन था उसमें आपको लैग फेज लॉग फेज ऑक्सीजन एक्सपोनेंशियल फेज स्टेशनरी फेज ऐसा डिक्लाइन फेज. और एक बार क्वेश्चन इसके ऊपर भी आया. तो logarithmic growth of bacteria means 10 raised to 10 10 raised to 20 10 raised to 30. So logarithmic increment in the bacterial growth that occurs in the following phase of micro following phase of bacterial growth cycle. लैग फेज लॉग फेज स्टेशनरी फेज डिक्लाइन फेज. तो मल्टीप्लिकेशन का होता है वो एक्सपोनेंशियल मतलब क्या 10 रेज टू 1 10 रेज टू ऐसा. ठीक है. तो एक्सपोनेंशियल मल्टीप्लिकेशन ऑफ बैक्टीरिया दैट ऑकर्स इन द लॉग फेज. Now bacteria multiplies by binary fission or by budding. If you want to read more about binary fission and budding, you can read from book. You can see over here, this is a chart where this is a graph. So if you plot a time on x-axis, growth of bacteria on y-axis, so you will get this graph. So this first is a lag phase. So it is known as a initial adaptation time. Okay. After that, exponential phase. So rapid growth of bacteria. So exponential growth, logarithmic growth, logarithmic or exponential growth of bacteria. After this, there is a straight line or there is a leveling phase. Leveling straight line. So why this is because there is a rate of multiplication is equal to rate of death. Okay? Death rate is equal to rate of multiplication. So that's why leveling. Then when the nutrition of your media is depleted, then the toxic metabolite will be accumulated. So at that time bacteria will start dying. That is known as the decline phase. ये phases याद रखो आप थोड़ा सा important है. Different staining technique. क्वेश्चन इसके ऊपर भी आता है आपका. Different staining technique. Staining technique बहुत ज़्यादा important है. So you have different staining technique. So for the identification of bacteria, how you could know what type of bacteria is there. यहाँ पे कौन सा है ग्राम पॉजिटिव है ग्राम नेगेटिव है एसिड पॉजिटिव है बेसिलाई है कौन सा है मतलब स्पोर रेडिंग बैक्टीरिया है या फिर एंडोस्पोर फॉर्मिंग बैक्टीरिया है कौन सा है. सो यू नीड टू आइडेंटिफाई द बैक्टीरिया. हाउ you can identify by two ways. First by staining technique, second way is there known as a there are two ways to identify the bacteria. First one is the staining technique and second one is the metabolic evaluation. Metabolic evaluation. Okay. So we'll see the first staining technique. So what are the different staining techniques? First one is a gram staining. So gram staining. gram ने क्या किया था, एक staining technique develop किया. So gram, known as a gram staining. So based upon this staining, bacteria are classified into two types: gram positive and gram negative. So those which will give you a color in the gram staining known as a gram positive bacteria, which will appear a purple or dark violet in the color, and gram negative bacteria will appear in the red in the color. First, in this staining, ये staining का पूरा प्रोसीजर कैसा होता है, क्या होता है, उसका इम्पोर्टेंट नहीं. So what is important in this staining? So what is the primary dye? What is the secondary stain? What is the counter stain? Which is the decolorizing agent? क्वेश्चन इसके ऊपर दो तीन बार. Remember, in the gram staining, crystal violet is a primary dye. Crystal violet is a primary dye. Okay, crystal violet is known as a primary dye or primary stain. So then after that, iodine is added. Okay. Iodine is serve as a mordant. Mordant का काम कौन करता है? Iodine. मतलब फिक्स करेगा वो कलर. Mordant. Iodine काम करता है mordant का. So crystal violet is the primary stain. Iodine is a mordant. Okay, mordant. Then alcohol is used as a decolorizing agent. Question इसके ऊपर आ चुका है. So decolorizing agent used in the gram staining. Alcohol is used as a decolorizing agent. And then saffronin. Saffronin is a counter stain. तो यहाँ पे देखो क्रिस्टल वायलेट को बोलते हैं प्राइमरी स्टेन. आयोडीन एज अ मॉडर्न बोल के यूज होता है उसमें. अल्कोहल डीकलराइजिंग एजेंट और सैफ्रॉनिन काउंटर स्टेन. तो आपको पहले क्या करना पड़ता है कि बैक्टीरिया क्या करो कि इनोकुल इसके बैक्टीरिया लूप पे ले लो और उसमें क्या आपका स्लाइड के ऊपर बैक्टीरिया आप उससे वहाँ पे इनोकुलेट करो. सो after that you have to wash that bacteria with distilled water, and then you have to add crystal oil in, you have to add iodine, and you have to add iodine, you have to add alcohol also and saffron. Okay. Now this is the procedure. Now you have to do this procedure half. First time you have to do this procedure in a half. आपको बैक्टीरिया इनोकुलेट करना है, स्लाइड पे लेना है, उसके बाद में क्रिस्टल वायलेट डालो, उसके ऊपर आयोडीन डालो. ओके. और देखो अगर उसको स्टेनिंग आता है पर्पल कलर का या फिर डार्क वायलेट कलर का स्टेन आता है, तो दैट इज नथिंग बट द ग्राम पॉजिटिव. अगर नहीं आया तो फिर क्या करने का, नहीं आया तो फिर अल्कोहल डालो, उसमें डीकलराइज करो. उसके बाद में क्या करो, सैफ्रॉनिन डालो. सैफ्रॉनिन इज नोन एज द काउंटर स्टेन. एंड उसके बाद में चेक करो माइक्रोस्कोप के नीचे. If that bacteria takes a primary stain, that is known as a gram positive bacteria, and if that bacteria takes a counter stain, that is known as a gram negative bacteria. So that's why gram positive bacteria will look like a purple or dark violet color. Gram positive bacteria look like a purple or dark violet color in the color because it takes the crystal violet. It gets stained by crystal violet. Gram negative. Okay. Now gram negative bacteria, it will not take crystal violet. Okay, that's why upon decolorizing with alcohol, so crystal violet will be removed. Upon addition of this counter stain, that is known as saffronin. Saffronin is taken by gram negative bacteria. Gram negative bacteria will appear like a red in color because they are stained by counter stain. Question इसके ऊपर ऐसा आता है. In the gram staining, gram negative bacteria will take following stain. वहाँ पे ऑप्शन आएगा क्रिस्टल वायलेट, आयोडीन, सैफ्रॉनिन, अल्कोहल, या फिर बोलेंगे व्हाट इज द रोल ऑफ अल्कोहल इन द ग्राम स्टेनिंग. इस तरह के क्वेश्चन आपको आ सकता है जीपैट में. ऑलरेडी एक दो बार आ चुका है क्लोराइजिंग एजेंट के ऊपर. डिपेंडिंग ऑन दिस स्टेनिंग बैक्टीरिया आर ग्राम पॉजिटिव और ग्राम नेगेटिव. Now second type of staining is nothing but the acid fast staining. Fast staining. Obviously this acid fast staining is acid fast bacteria, particularly Mycobacterium tuberculosis and Mycobacterium leprae. So this carboflucin is the acidic dye. This acid fast staining is used for tuberculosis bacteria and leprous bacteria, that is Mycobacterium tuberculosis and Mycobacterium leprae. So carboflucin dye, which is the red in color, it is used. Okay, along with the acidic alcohol. Carboflucin, a red dye along with the acidic alcohol is used. Out of that, this alcohol will serve as a decolorizer. If bacteria is acid fast, then it will get stained. Color उसका color आ जाएगा. So acid fast bacteria get red color Non-acid fast bacteria will get colorless, will look like a colorless. Non-acid fast bacteria will not be stained by this carbofluorescence, but acid fast bacteria will be stained by this carbofluorescence. इसके ऊपर क्वेश्चन आता है कि which one is the dye used in the acid fast staining? Carbofluorescence. Carbofluorescence. Carbo, यार नाम याद रखो. Carbofluorescence. Now next, and this acid fast staining is used for Mycobacterium, Mycobacterium tuberculosis, Mycobacterium levi. क्वेश्चन इसके ऊपर भी आ चुका है डिफरेंट एग्जाम में, मतलब फार्मासिस्ट एग्जाम या फिर आपका डीआई के एग्जाम में उसमें. नेक्स्ट टाइप ऑफ स्टेनिंग इज झील निल्सन स्टेनिंग. झील निल्सन स्टेनिंग. The Thiel Nielsen staining is used for Mycobacterium tuberculosis. Okay. So remember, ऐसा क्वेश्चन भी एक बार आया था कि one of the following staining technique is not used for depression of Mycobacterium. उसमें Thiel Nielsen staining and acid fast staining उसके लिए होता है. So remember, Thiel Nielsen staining is also used for Mycobacterium tuberculosis. Now, whatever smear, smear means bacteria on your slide is rinsed with the acid alcohol mixture and solution of carbofluorescence. The results are same like that of acid. में difference क्या है? Acid fast staining में और Thiel Nielsen staining में सिर्फ मेथड का डिफरेंस है. आप कौन सा पहले अप्लाई करते हो, कौन सा बाद में ऐड करते हो. So there is no difference. Only बाकी मेथड का डिफरेंस है. बाकी रिएक्टर मतलब डाई सेम ही है. कार्बोफ्लोरेसेन इसमें भी कार्बोफ्लोरेसेन है. The method of application is different. So it is used for staining of Mycobacterium tuberculosis. Smear is rinsed with the acid alcohol mixture and solution of carbofluorescence is added. Now, if bacteria is acid fast, then it will get stained. It will get stained like red. रेड कलर आया तो एसिड फर्स्ट है, अगर नहीं आया तो नॉन एसिड फर्स्ट है. नेक्स्ट वन इज कैप्सूल स्टेनिंग. इज आल्सो नेगेटिव स्टेनिंग फॉर कैप्सूल. क्या है? इट इज आउटर कोट और आउटर एनवलप प्रेजेंट ऑन द बैक्टीरियल सेल. बैक्टीरियल कल्चर आर मिक्स्ड विद द निग्रोसिन डाई. बैक्टीरियल कल्चर इज मिक्स्ड विद द निग्रोसिन सॉल्यूशन, इंडियन इंक सॉल्यूशन, और इंडियन निग्रोसिन डाई यूज करते हैं या फिर इंडियन इंक डाई यूज करते हैं. नाउ सैपरानिन इज एडेड. कैप्सूल बीइंग ए डिफरेंट इन केमिकल स्ट्रक्चर composition, they are not stained by biological dyes. कैप्सूल क्या है? कैप्सूल का नेचर क्या होता है? कैप्सूल का स्ट्रक्चर थोड़ा सा अलग होता है. They are not stained by biological dyes like this nigrosin. Capsules, therefore, most of them appears as a whole just around the stained microbial cell. कैप्सूल का स्ट्रक्चर और माइक्रोबियल सेल का स्ट्रक्चर थोड़ा सा डिफरेंस होता है. कैप्सूल स्टेन नहीं होता है, वहां पे सिर्फ माइक्रोबियल स्टेन होता है. माइक्रोबियल सेल ही कलर उसमें आ जाता है. सेल कोई माइक्रोबियल सेल कोई कलर आ जाता है, कैप्सूल को नहीं आ जाता. कैप्सूल विल लुक लाइक अ होल्स just surrounding the colored microbial cell. Okay, that is known as a capsule staining. Now next one is the endospore. You have to be little bit cautious, you have to be little bit serious because you have only 4 months and you have to cover up a lot of syllabus. Now next one is the endospore staining. Endospores. So for that special staining is there known as Schaffer-Fulton endospore stain. Schaffer-Fulton endospore stain. इसके ऊपर क्वेश्चन आ चुका है. Schaffer-Fulton endospore stain. Endospore का टर्म नहीं देंगे वहाँ पे, वो सिर्फ बोलेंगे Schaffer-Fulton stain is used in the following type of staining technique. Endospore stain. So endospores they are little bit resistant to staining. वो रेजिस्टेंस होते हैं स्टेनिंग के लिए. सो दैट्स व्हाई यू हैव टू हीट द बैक्टीरिया. बैक्टीरिया को इनकल करो आपके ग्लास स्लाइड पे, उसके बाद में उसको हीट करो, फिक्स करो. बिकॉज ऑफ हीटिंग व्हाट हैपेंस, द डाई कैन गेट पेनिट्रेट इनटू द एंडोस्पोर. ओके, सो एंडोस्पोर इज हीटेड विद टुगेदर विद द मर्जेंटाइट ग्रीन डाई. It is washed to clear the dye and finally counterstained safranin is added and they are seen in under the microscope. So endospores are stained green in color. याद रखो. टेम्परेचर किसमें यूज करते हैं? एंडोस्पोर स्टेनिंग में. नेक्स्ट वन इज अ फ्लैजेला स्टेनिंग. So carbofluorescence and modern is used to process the flagella staining. Now this flagella staining is a tedious process. flagella को स्टेन करना थोड़ा सा कठिन होता है. So I don't know whether you have done this staining technique or not. जब हम कॉलेज में थे तो हमने सब किया था. ग्राम पॉजिटिव मतलब ये सब ग्राम स्टेनिंग किया था, ये कैप्सूल स्टेनिंग किया था, सॉरी ये एंडोस्पोर स्टेन भी किया था, एसिड फास्ट स्टेनिंग किया था. डोंट नो वेदर यू आर डूइंग और नॉट. आता है उसको. नाउ ग्राम स्टेनिंग एंड एसिड फास्ट स्टेनिंग दे आर नोन एज अ डिफरेंशियल स्टेनिंग टेक्निक। ग्राम स्टेनिंग जो है और एसिड फास्ट स्टेनिंग जो है उसको डिफरेंशियल स्टेनिंग टेक्निक बोलते हैं। ये लाइन एज इट इज याद रखो सुना। Difference between gram positive and gram negative bacteria. Difference between gram positive and gram negative bacteria. So as I have told you, gram positive bacteria and gram negative bacteria, they will have a different composition in the cell wall. GPAT 2021 में question as it is आया था यहाँ पे इसके। We'll see what is the difference in gram positive gram negative. First of all, first point is nothing but the cell wall component. कि cell wall किसका ज़्यादा है किसका कम है? Cell wall thickness. First, cell wall thickness is more thick in case of gram positive bacteria. So it is around 20 to 25 nm in the thick nanometer. 20 to 25 nanometer होता है gram positive में. तो gram negative में कितना है? 10 to 15 nanometer. So remember, cell wall is thicker in gram positive. It is a thinner in gram negative. Then peptidoglycan layer. Peptidoglycan layer. It is a component of cell wall. Okay. So peptidoglycan layer is more in case of gram positive. It is around 50 to 90%. It is a less in case of gram negative, around 5 to 10%. And that's why penicillins they are mostly effective in case of in case of gram positive. Gram positive bacteria में peptidoglycan का layer जो है वो ज़्यादा quantity में होता है 50 to 90%. And that's why penicillin they can bind with the peptidoglycan. And the cross linking of peptidoglycan and penicillins, okay, sporins, penicillins. That's why penicillins, cephalosporins, antibiotics, they are not effective, much effective in case of gram negative because gram negative will have thin, it will have a less peptidoglycan layer. Okay, remember. Another component is nothing but the teichoic acid. Teichoic acid. So, question there was a question in GPAT 2021. Teichoic acid is present in the one of the following: positive, gram negative ऐसा option में gram positive bacteria will have teichoic acid. Teichoic acid is present in case of gram positive bacteria. It is absent in case of gram negative bacteria. Polysaccharides they are present. Again, polysaccharides are present in the gram positive bacteria, absent in the gram negative bacteria. Lipid content. सेल में सेल वॉल में लिपिड कितने हैं? So less lipid content or absent. So lipid content is very less or it is absent sometimes in case of gram positive. More lipid content is there in case of gram negative. Nature of cell wall. So, nature of cell wall. Cell wall is simple in case of gram positive bacteria. Cell wall is very complex in case of gram negative bacteria. Effect of lysozyme on cell. What is lysozyme? Lysozyme क्या होता है? So these are the enzymes. These are the enzymes produced by various cells. For example, जैसे टीयर्स में क्या है? आपका जो टीयर होता है, आंसू जो होता है आपका आंख से निकला आंसू या टपका का शराब में कुछ भी उठाना है. दैट आंसू आपका जो टीयर होता है उसमें भी क्या होता है कि लाइसोजाइम एंजाइम होता है. So lysozyme enzyme they can digest the bacteria. So effect of lysozyme on cell. So easily digested or easily destroyed. Gram positive bacteria is easily destroyed by lysozymes. Gram negative bacteria is distant. Gram positive bacteria जो है is easily destroyed by your lysozyme. तो आपका जो टीयर होता है टीयर आंसू उसमें भी lysozyme enzyme होता है. So they digest the microbe. Gram positive bacteria is easily destroyed or digested by the lysozyme. Gram negative bacteria they are resistant. Streptomycin and tetracycline susceptible है कि नहीं? Gram positive bacteria is less susceptible for tetracycline and streptomycin. मतलब tetracycline streptomycin अगर देंगे और उसको gram positive का इन्फेक्शन हो गया होगा तो वो क्लियर नहीं होगा. Because gram positive bacteria is less susceptible for streptomycin and tetracycline. और gram negative bacteria is susceptible. मतलब streptomycin tetracycline gram positive को नहीं मार सकते, gram negative को मारेंगे. gram positive को कौन मारेगा? Penicillins and cephalosporins. इसको क्या कौन मारेगा gram positive को? Penicillins and cephalosporins. ठीक है? Because penicillin cephalosporins act on a cell wall where there is a large amount of peptidoglycan that is present in the gram positive bacteria. Penicillin and sulfonamide susceptibility. So obviously I told you penicillin, okay, gram positive bacteria is susceptible for penicillins as well as sulfa drugs. Now gram negative bacteria it is less susceptible for penicillins and sulfur. Okay. Now इसके ऊपर एक बार क्वेश्चन आए examples. What are the examples of gram positive bacteria? नाम आपको याद ही रखना है, इसके लिए कोई ट्रिक नहीं. So remember Bacillus anthracis, Clostridium tetani, Staphylococcus aureus, then Corynebacterium diphtheria. So these are the examples of gram positive bacteria. Bacillus anthracis वो anthrax करता है आपको. Bacillus anthracis will cause anthrax disease. Clostridium tetani will cause tetanus. So that is a gram positive. Staphylococcus aureus, most of the respiratory tract infection. It is caused by Staphylococcus aureus. RTI, respiratory tract infection. ये aureus से होता है. Corynebacterium diphtheria causes diphtheria. So all these are the examples of gram positive. Now what is the example of gram negative bacteria? Remember E. coli, Salmonella typhi, Vibrio cholera, Haemophilus influenza. These bacteria are known as a gram negative. So question आ चुका है, one of the following is not a gram positive bacteria. Coli is not a gram positive. Salmonella is not gram positive. Cholera is not a gram positive. Influenza is not gram positive. Okay. क्वेश्चन इसके ऊपर दो बार क्वेश्चन आ चुके हैं. टिकोइक एसिड इज नॉट प्रेजेंट इन ग्राम नेगेटिव बैक्टीरिया. सेल वॉल इज मोर थिकर इन ग्राम पॉजिटिव बैक्टीरिया. इसके ऊपर भी क्वेश्चन आता है. पेप्टिडोग्लाइकन लेयर इज प्रेजेंट इन द हाईएस्ट the following. Identify the wrong statement about the gram positive gram negative bacteria. ऐसा भी क्वेश्चन आएगा, या फिर आपको मैच द फॉलोइंग का भी क्वेश्चन आएगा. सिंगल चार्ट के ऊपर बहुत सारा क्वेश्चन आ सकता है. नाउ बैक्टीरियल स्पोर्स. Many bacteria produces spores in the cell. Many bacteria produces spores inside the cell. अगर बैक्टीरिया कोई स्पोर्स बनाता है inside the cell, that is known as endospores. Many bacteria produces a spore inside the cell known as endospores. Now, if bacteria produces the spore outside the cell known as exospores. Okay. So, for example, those bacteria which produces the spores, they are known as a sporulating bacteria. जो बैक्टीरिया स्पोर्स बनाते हैं उसको बोलते हैं sporulating bacteria. For example, Bacillus anthracis, Bacillus subtilis, then Clostridium tetani, Clostridium botulinum, then Coxiella burnetii, then Thermoactinomyces, Streptomyces. These are known as sporulating bacteria. ये स्पोर करते हैं. स्पोर क्या है? स्पोर विल हेल्प द बैक्टीरिया टू सर्वाइव इन द एक्सट्रीम कंडीशन. तो स्पोर का जो स्ट्रक्चर होता है वो बहुत ही हार्ड होता है. उसमें यू कैन नॉट किल द स्पोर बाय ऑर्डिनरी स्टरलाइजेशन मेथड. दे आर रेजिस्टेंट टू हीट, दे आर रेजिस्टेंट टू मॉइस्ट, दे आर आल्सो रेजिस्टेंट टू केमिकल डिसइंफेक्टेंट. जो स्पोरुलेटिंग बैक्टीरिया है वो उसको मारना बहुत ही मुश्किल है. Formation of bacterial spore is known as sporulation, and it is the method of preservation of bacteria, not reproduction. ठीक है. इससे क्या होता है कि स्पोर क्या करते हैं कि बैक्टीरिया को प्रिजर्व करते हैं. Spores are extremely resistant to desiccation, staining, disinfection by chemicals, then radiation and heat. Now spores can be remain viable for centuries. कुछ 10, 20 साल, 50 साल, 100 साल तक बैक्टीरिया जिंदा रह सकता है उसमें. Okay, spores can remain viable for centuries and help bacteria to survive for longer period under the extreme condition. Extreme means non-stable condition. एक कंटेंट होता है, इसके ऊपर क्वेश्चन आ चुका है. Spores of the most of the medicinal important bacteria, they are destroyed by moist heat sterilization, that is autoclaving at 121 degrees Celsius for 20 minutes, heating for 2 hours at 160 degrees. Spores of the most of the medical important bacteria that can be destroyed by moist heat sterilization, that is autoclaving at 121 degrees Celsius for 20 minutes, or by using dry heat. So you have to heat 160 degrees Celsius for 2 hours. All the spores होते हैं उसमें एक केमिकल होता है. So all spores consist of a large amount of dipicholinic acid. Because of that dipicholinic acid, bacteria is resistant for sterilization. All spores consist of a large amount of dipicholinic acid with, okay, when this combines with the calcium, forming calcium dipicholinic complex. Now this calcium dipivolinic complex plays a very important role in the heat resistance of bacteria. जो एंडोस्पोर्स जो होते हैं वो हीट रेजिस्टेंस क्यों होते हैं? Because of this calcium dipicolinic acid. It makes bacterial spore resistant to heat. Okay, calcium dipicolinic acid क्वेश्चन इसके ऊपर आ चुका है. Okay, so spores consist of following: dipicolinic acid. And for staining of endospore, Schaffer-Fulton method is used for the staining of Schaffer-Fulton method. क्वेश्चन आ चुके हैं इसको. Remember, spores can be eliminated by 121 degrees Celsius at 20 minutes by autoclaving, and it can be removed by heating the solution, heating the whatever in the sample or container, heating it at a 160 degrees Celsius for 2 hours. 160 डिग्री तक हीट करना है दो 2 आवर्स के लिए, तब जाके स्पोर्स डिस्ट्रॉय हो सकते हैं. फिर ऑटोक्लेव करना है 121 डिग्री सेल्सियस फॉर 20 मिनट. Spores consist of a dipicolinic acid. When they combine with the calcium to form a calcium dipicolinic acid complex, which plays a very important role to make the spore resistant to the heat. Now, Schaper-Pelton method is used for the endospore staining. Now, microbes are differentiated on the basis of various catalyzed metabolic reactions. Presence of presence or absence of certain enzymes, intermediate metabolites, they give the information regarding the identification and classification of microbes.